Okay, in video 1 and 2 of this tutorial, I showed you how to process multiple samples, or 2 in our case, with Kraken 2 and Bracken, and how to generate aggregated tables that contain either the relative abundance or the total number of row counts for the samples that we have processed with Kraken 2 and Bracken using a Python script. But this didn't show or give us an opportunity to check the full lineage of the taxa ID for which we had aggregated tables with relative abundance or total number of reads. So what kind of tool can we use in order to obtain the full taxonomic lineage for each taxa and to generate aggregated tables at the same time? So the tool is called taxpasta. So I have the commands that are required in the Github repository for us to use taxpasta. So the first important thing that I need to mention is that taxpasta works with Python 3.8. And the content version that I have at the moment, the default Python is, let's check it, version is 3.7. So what I can do, I can create a new content environment where I specify the Python version that we need in order to install taxpasta and to run it. And this is exactly what I'm going to do. And this is exactly what I have already outlined in the tutorial. So we're creating environment that is called my environment, not super creative name, but it doesn't really matter. And then we specify that we need Python version 3.8 to be installed together with this environment. Then we activate the environment, and then we install taxpasta. I prefer to install it with pip because when I try to install it with conda, I have some problems with dependencies that are not matching. And with pip, there was no problem at all to install. So this is my preferred way of installation. Another thing that we will need in order to run taxpasta is to download some files that are going to be required if we want to append the full lineage to the aggregated reports. I suggest that you make a new directory. So we go out of our working directory, and I think I call it taxa_db. So you go into this directory, you have to create it, of course, just follow the instruction in the tutorial. So make directory, when you go out of your working directory, cd taxa_db. These are just a few commands that I copied directly from the menu of taxpasta. And what they will do, so we're going to download a bunch of, just one target zip file, then we unzip and dearchive. But in the meantime, we also create a new subdirectory that is called taxdump. And if we want to check what it is present in the taxdump folder, all of those files. Let's have a look. I have already done that. What do we have here first? If you go in the taxdump folder, lsla, the same files that are listed in the GitHub repository. So the full path to this directory is this, Combalino taxa_db taxdump, and we will need this full path when we run the taxpasta command. So the difference between the Python script that I showed you in video 2 and taxpasta is that they use different bracken outputs. So the inputs for the taxpasta are the TSV files and not the bracken reports as we previously saw for the Python script. So in addition to the input files that are the outputs from bracken, we specify the argument at lineage, which means that we would like to add the full lineage along with the aggregated reports that are going to be generated from running taxpasta. So by specifying this argument here, we tell taxpasta that we would like to get aggregated reports. Here, this is the name of the program that used to generate the input files that we are using, and in this case it's of course bracken with O, just the name of the output file, aggregated output file that's going to be generated. Taxonomy argument is the full path to the folder that contains all of those D and P files that are going to be needed for appending the full lineage. You need to go back to your working directory. In our case, that was the fastq random subsample one. And you have to run here this command, copy and run it. And once you do, you will have this output generated. Let's have a look. Okay. But I don't think that this was the best way to check the output. So maybe let's do that with Excel. So I already have it open in Excel. So we have taxonomy ID, which is corresponding to the tax ID from the aggregated files that were generated with Python. This is good. Then we have the full lineage, you see? So we have bacteria, Terrabacteria, Actinomycetota, Actinomycetes, so on and so forth until we reach the level of the species. Then the results for sample 1 bracken and sample 2 bracken, so these are the TSV files. Here they are not in relative abundance, they are present in row counts for each of the samples. To be honest, I don't really know how to obtain relative abundances directly from taxpasta, but an easy thing to obtain relative abundances with the full lineage is simply to merge the output that you have from the Python script with the relative abundances with this file, and you will have to merge on the columns of taxonomy ID from here and tax ID from the output from the Python script. Okay. And another important thing that I have to mention is that when we run the analysis with taxpasta, because we specify the TSV files, which are really the output from the taxonomic level that we acquired when we ran bracken, we do not obtain the results for all different taxonomic levels. So here we only get the results for species. If we would like to get the full lineage, for example, for genus, we will need to generate new TSV files, rerunning bracken with the L argument G, and then we will need to perform the same additional steps with taxpasta on the TSV files that contain the information for genus. But having all these steps in place, by far, you should be able to generate files that contain all information necessary for downstream processing, such as identification of differentially abundant taxa across groups. There are quite a lot of programs are packages that can perform this type of analysis. So let me know in the comments if you'd like me to probably show you how you can do that in another tutorial. I hope everything was clear. So the Python script is in the GitHub repository as well. All these files, the output files and the intermediate files are also there, so you can open them and have a look at them. Yeah, this is from me for now. I hope it was useful. Bye bye.